All right. So for this week, we are going to be going over natural selection postulates, um, life history evolution, intro to genetics, and Hardy-Weinberg equation. So we're kind of working our way still through evolution. We'll leave it for just a minute to pick up some genetics background so that we can then go to uh, kind of looking at evolution from a genetics point of view. So that's kind of the where we're at. Um, all right. So for natural selection postulates, there are four main postulates that are part of natural selection. First is that there must be variation uh, in the in the population. Okay, and that variation ultimately comes through random mutation. Now, this is one key common misconception that students have, people in general have about how evolution takes place. There is not a need that arises, some sort of limiting factor that comes up that then results in a mutation occurring to deal with that limiting factor or problem. Rather, there are mutations that exist and therefore variation, right? There's the different colored dots from our simulation. Then natural selection can act upon those differing dots. If they were all the same color, if there was no variation, then that postulate of natural selection would not be met, and we couldn't have natural selection and therefore evolution occur because there's only one type of dot. There's no variation. So there must be variants in the population that are better at dealing with a particular limiting factor. Now, as natural selection continues, you can have that mutation be compounded or become even stronger as selection takes place, but the initial difference has to be there to start with. And so that's our first postulate. So when you're thinking about this, if there is no variation due to mutation, there can be no natural selection. So when we're thinking about postulates, these are things that must be met in order for natural selection to occur. Our second postulate of natural selection is heritability. So individuals must be able to pass, so there has to be differences in the population, and those differences must be able to be passed on to the next generation. If that's not the case, we don't get natural selection. We get differences in who lives and who dies, but those differences don't get passed on to offspring. And so we don't get a shift in the population. The idea is that the next generation will be more like those variants that are best able to deal with the limiting factors. So we have to have differences, and those differences must be heritable, must be able to be passed on. Next, we must have a population that has the ability to reach their biotic potential. Essentially, they have to be able to reproduce exponentially. They have to get to a point where that carrying capacity comes into play, where that carrying capacity starts to matter. If they can't ever get to that point, there's just not going to be natural selection that occurs because there's never going to be competition that makes it matter that there are differences. So there has to be an ability to reach your biotic potential, and there has to be limiting factors in the environment. So we have to have biotic potential. That's one postulate. And then another postulate is that there is limiting factors. So there is a predator or disease or ability to acquire food or shelter. Those things have to be in existence. And those limiting factors establish a carrying capacity, which then their biotic potential takes them to. And that's when we get the competition that we need. So there must be limiting factors for natural selection to occur, and there must be biotic potential. They must have approached that point. Finally, there must be some individuals that have a greater fitness than others. Fitness in this case does not mean physical prowess. It doesn't mean physical characteristics necessarily. What it means is an evolutionary fitness. How fit are you for the environment and the associated limiting factors that are coming into play there? The greater fitness is who survives and the number of offspring that they produce and the number of offspring that survive. That's kind of how we measure fitness. It's how many, the reproductive success of you and your offspring are essentially how we measure fitness when it comes to evolution. So we have to have variation. That variation must be heritable. That variation must make a difference. There must be some form in that variation, in the differences between individuals in our population. There must be a variant that helps an individual have greater fitness. If none of the variants help with that, we don't get natural selection. So for example, like you have attached earlobes and unattached earlobes. Having attached earlobes and unattached earlobes, there's no evolutionary advantage to that in humans. There's no greater reproductive success that comes from that. So in that case, we would have variation. That variation is heritable. We might even be approaching our carrying capacity due to limiting factors. But because there's no, that particular characteristic doesn't make a difference in reproductive success, we're not going to have natural selection take place. So we have to have differences. Those differences must be heritable, and those heritable differences need to make a difference in the evolutionary fitness of individuals within the population. So that's the main thing from our natural selection postulates. Kind of an interesting way of looking at how sexual reproduction could become evolutionarily advantageous, right? And primarily that's what we see in these flowers that could they could asexually reproduce versus sexually reproduce with another flower of a different plant, right? And what we see is the number of offspring that survive are much higher. That means that evolutionary fitness is increased by sexual reproduction among these flowers. And we actually see that repeated over and over again. That we'll get to in a future lesson. But just kind of a nice example of what is evolutionary fitness when we look at measuring it in this system, and how it's not that sexual reproduction itself could become an evolutionary trait that is advantageous, and then natural selection would favor that. And we see sexual reproduction in most species, so that's a good indicator as to why. All right, any questions on natural selection postulates?
Yeah, Professor, I had a quick question.
Yeah.
As you were kind of talking about whether or not it helps with our ability to reproduce and whatnot, I was just kind of thinking, nowadays is this as prominent? Say like, obesity is like I mean, it's obviously a disadvantage for finding a mate, but also it's just a disadvantage for living a long life. But it still is a pretty big problem. Do you think that just our
So
current circumstances, or what do you think about that?
So there's a few things that can come into can impact us here, right? When we're thinking about that. First, is that strictly genetic? I would say no, right? That there's a significant how you live your life, environmental factor that plays a role there. So the heritability of that can be a little bit iffy. There is definitely some genetic component to it, but it's not as strong as environmental. So whether evolve, so that makes it tricky to evolve there. A second thing is that a lot of the truly negative effects on that, right? Your lifespan, for most people, those things are going to take place around 50, 60, right? That they're going to have a shorter lifespan. Most people are done reproducing at that. And so the shorter lifespan doesn't actually reproduce or result in a lower reproductive success. And then as far as like acquiring a mate, I think that may be the case. I'm not entirely sure though. I'd really want to see that is actually the case. because obese people can find other obese people, for example. So it's a little bit tricky, I think, even on the reproductive
That's an
angle.
excellent answer. Thank you. That's satisfactory.
Okay. As far as like humans, though, right? Like a lot of the things that prevent us from evolving due to natural selection would be the limiting factors component, right? What is our carrying capacity is something that we don't really know, and so we're able to help people survive that have conditions that wouldn't have been able to survive in the past. Um, there is some natural selection that takes place but those are typically going to be things that are happening in third world countries. HIV has caused natural selection on human populations in Africa, for example. But it's probably not going to be widespread and as common as you might see in animal or plant populations. So, good question. All right. So next, we're kind of looking at life history traits, and this is kind of take us from that idea of sexual reproduction and go a little bit further there. So life history traits, there's three components to that. How essentially how an organism is born, lives, and dies. Okay, and how it goes through that. And so the model that we've created has these three components to it: growth, maintenance, and reproduction. So growth meaning how quickly do you reach an adult size, how long it takes, how big you are, all those things kind of fit into growth. Maturity, all of that's there, okay? And then we have maintenance. This is essentially how long can you live? So how do you keep yourself alive long term? And then we have reproduction, which is how often do I reproduce? How many offspring do I have when I reproduce? And the idea is, after they came up with this model, is they said, well, why don't we see evolution favoring all three of these things at once? Why don't we see the hypothetical Darwinian demon, right? Something that is excellent at all of these aspects of life. And as you go through this lesson, hopefully you're able to see that the reason why is because environments aren't all the same. And life isn't predictable. If you have a high predation environment, waiting to reproduce could mean that you don't have any offspring at all before you're eaten. In that case, it's better to reproduce early in life. In contrast, if you have a low predator environment and you have very stable conditions, it's better to put off reproduction. And you can have members of a population get split into very locally, like geographically close, but very different systems due to things like predators or disease being present. And as a result, you can have very quickly, say, two streams with a type of fish in each stream that used to be one population. Now, all of a sudden, this fish, because it has a high predation factor in its environment, is going to be reproducing at very young ages and having very small overall sizes. And this group is going to reproduce later in life and get to be very large. And within just a few generations, depending on how strong that predation factor is, you can see a shift to where these species may not look similar at all and may reproduce at different times. And so this understanding that there is no ideal in these three factors, because environments are different, can help us understand how all of a sudden species form, right? Speciation takes place. And we'll come back to that later. But that was one of the big things, is that essentially there's a different answer for what's best depending on the environment. There was also another component to this, is that there seems to be trade-offs, right? That if you're going to maximize reproduction, you can't, for whatever reason, through biological constraints, right? The way that the organism grows and matures, it can't maximize growth if it's going to maximize reproduction. There's a trade-off to it. And so that's how they come up with this model, right? That's great and explains things. And then they come to this question of, well, why don't we see something doing great at all of these things? And that's when, well, we have to be able to explain why we don't see that. And that's where a lot of these experiments have come back to, is whenever one goes up, reproduction here or maintenance goes up, growth, the other comes down, right? So we have trade-offs in in this model, and that best is what we think best explains the pattern of organisms, or in the case of the Darwinian demon, that we don't see. So the part I wanted to get to, talking about speciation, right? That there are ways that species become isolated, genetically isolated from one another. Now, if they're genetically the same, that means that they're part of one population and the members of that population are able to reproduce back and forth. It's called the biological species concept. They're able to essentially breed back and forth and produce viable offspring. If they, as they're going through that process, or that population is going through, there's different ways that they can become isolated to the point where they can no longer reproduce with one another. So an example of that might be what we call behavioral isolation, that these two became separated for some reason. And at some point, this group on the left here starts having a different behavior, like maybe a song. Songs can be really important for mate selection in birds. This group is singing the song slightly differently than this group. And because of that, whenever they meet in their in-between zone, right? If they were ever to have individuals come across each other, they aren't singing the right song. And so they can become behaviorally isolated. Probably the most common way that groups can become genetically isolated is they are just physically in different geographic locations. And in that case, they can't reproduce, and differences, then all of a sudden they're on separate evolutionary paths. So we're thinking of this with like our evolutionary trees. This is the split, right? The population gets geographically isolated or behaviorally isolated. Another one is temporally isolated. And that fits kind of with what we were talking about with, like, in certain environments, it's better to reproduce early versus late. And in others, it's better to reproduce late than early. And in that case, right, you're going to not be able to reproduce because the females are not receptive at the same time. So an example of this might be like elk and deer, right? Elk here in Utah are in their reproductive stage, kind of late September, early October, whereas deer are in their reproductive phase in November, right? So because of that temporal isolation, those two groups are reproductively isolated. So these are all what we call pre-zygotic isolation. So before the organisms attempt to reproduce. There are others that are post-zygotic, right? So the sperm and egg can fuse together to form the zygote, but then the zygote is not viable. So it just will never, the developmental plans are so distinct that they won't ever form an organism. Sometimes they do form an organism and that organism is born. So a classic example of this, horse and a donkey reproduce to make a mule, but a mule is typically sterile, right? So hybrid sterility can result in these isolations. So this life history stuff is directly tied into how do these organisms develop these isolations, and a lot of it has to do with being in environments that have these different pressures. So, all right, any questions on life history evolution? Okay. All right. Next, we have the intro to genetics. We're just going to make sure we get some key terms down here, and they're bolded in the lesson, but I just want to talk through them and give you a different. So a gene is a segment of your DNA that codes for a specific what we call phenotype or appearance. So the gene is responsible for, there's a whole fields of science that are about how do we go from the gene to the protein. And we'll go into that a little bit later in the semester. But we'll just keep it simple and say a segment of DNA that codes for a physical trait or a trait in some way. And that is what we call phenotype. Alleles are differing forms of that same gene. So for example, we could think of a gene as a segment of DNA is the recipe for a cookie. And I can have an allele that calls for chocolate chips to be put into the cookie. So the genes give me the cookie, then I have the form that gives me chocolate chips. And then there are some sick, twisted individuals out there who like to make others feel really bad, who put raisins into cookies. So that's a different allele. So we have the chocolate chip allele and the raisin allele. Both of them are giving us a cookie. That gene codes for cookie. I could have the chocolate chip version, the one that everyone should want, or the raisin version that's a lie. And you, so that's the idea of the relationship between gene and allele, right? Gene is, we have a recipe for a cookie, and then I have these different forms of that cookie that I can produce, which would be the alleles. Each individual that are part of a diploid organism, like humans are, diploid means we have 2 copies of every gene. So I could have a copy of the chocolate chip recipe or the allele, and I could have a copy of the raisin, right? I could have 2 chocolate chip alleles. I could have 2 raisin alleles. So I have these different, what we call genotypes, these different patterns of it. In the fruit flies, we had red eyes versus dark eyes. And so the red allele and the dark allele are there. And you could have 2 red alleles, you could have 2 dark alleles, or you could have the in between. If you have 2 of what we call the dominant allele, a dominant allele means that if I have one of each, that's a heterozygous genotype. The genotypes are the sets of alleles that you have 2 of each, right? So a heterozygous individual, for if they have a red eye and a dark eye allele, will have a red physical appearance, a red phenotype. That tells us that this is the dominant allele because it shows up versus the recessive allele, which does not, when both are present. So the dominant allele and the recessive allele, when they're together, that's how we know which one's dominant and recessive. If I just have 2 of the red eye alleles, that's called homozygous. If I know that it's dominant, I can call it dominant homozygous, but I don't know that until I've seen an individual that I know has one of each. I also can have 2 of the dark eye alleles, so that would be homozygous recessive. So we have phenotype, which is what's the physical appearance, right? The dominant physical appearance or the recessive physical appearance. Then I have genotype, where I can be homozygous dominant because I have 2 of the dominant alleles, heterozygous because I have 1 of each, or homozygous recessive because I have 2 of the recessive alleles. So we want to be familiar with that terminology. So the question here is, so are alleles just types of genes? Yeah, they're a form of a particular gene, right? So eye color is the gene, and you can have the red allele or the dark allele. So it's differing forms of the recipe for eye color. Terminology here. One thing that's important to keep in mind is that you have 2 of each allele 2 alleles of each gene. When you go to reproduce, you only get to pass on 1 of your 2 to a particular offspring. You can't pass both of them on to the same offspring. So that's the law of segregation. We must segregate your 2 alleles. Now, we have no way of knowing which of your 2 will go to a particular offspring. It's a random chance. And so when we're setting up a genetic problem, when we're doing a Punnett square, the reason why we put both of your alleles on there is because both are equally possible of being passed on to the offspring. And so if you were heterozygous and you had a red allele and a dark allele for eye color, we have to set up the Punnett square where you can pass on. That's why we have the 2x2 box, right? So we have parent one, they could pass on the big R or the little r. And then we have parent two, who's going to be the vertical axis, who could pass on the big R or the little r if they're both heterozygous. And we have to do that possible combination. So the law of segregation says we need to separate those as we go to form an offspring. We can only pass one of them on, and that's the same for both parents. And so when we're doing a Punnett square, we're really just setting up doing all the possible combinations that a pair of individuals could produce. Yes, Austin, go ahead.
So using eye color as like the gene example, what if there's like, I mean, there's certainly more than two eye colors. So is the idea in every single person, you can only possess two different types of eye color that you could potentially pass on?
Correct. And human eye color gets a little bit tricky. So it's actually, there's not one gene that controls it. There's multiple genes that control it. So the first gene, we'll think of this as, do you put pigment in your eye? Okay. If you do, then we go on to the second gene that controls which color it is, right? Whether you get kind of hazel eyes or green eyes or brown eyes. But if you don't have the dominant allele, if you have two recessive alleles for that first gene, you don't get any pigment. And the color that shows up if you don't get pigment is blue. So eye color is a little bit tricky in that case. So hopefully you kind of see how that like kind of builds on itself, right? First gene, do I get pigment? If I don't get pigment, I get blue eyes. If I do get pigment, then it goes on to the second gene, which then there can be more than one or more than two alleles, right? I can have an allele for green eyes, blue eyes, hazel eyes, 3 alleles, right? Now, as an individual, I only get 2 of those. But there can be more than 2 options for the 2 that I get.
Yeah, that makes sense. So you would never have like an uppercase R, lowercase r, cursive R or something. I mean, it's only gonna be 2, right?
Yeah, just be the
Okay.
2 that you end up with.
Okay, thank you.
Okay. The next part that I want to talk about is the law of independent assortment. And what we see here is that we have, instead of just one trait, we're looking at eye color and fruit flies. We're also looking at wing shape here. So eye color and wing shape together. Now all of a sudden that's a little bit trickier, right? Because we're not looking at just 2 alleles for each individual. We're looking at 2 alleles for eye color and 2 alleles for wing shape. And those 2 are not tied together necessarily. They independently assort from one another. That means that, let's say these top 2 here are our parents here, okay? The big R from this parent can go to either of these 2 big ends. And this big R could go with either of these 2 ends. So when we're making our Punnett square here, we have to do all the possible combinations. So for example, this third genotype here, we could have when we're forming the gametes, when we're making sperm or egg, this individual could pass on the big R and the big N together. They could pass on the big R and the little n together. We're still doing the law of segregation, right? You're only getting one R and one N, but we don't know which ones are going to be paired together in that sperm or egg. You could have the little R with the big N, little R with the little N. So because of that, the way that this third genotype plays out, you could have 4 different types of gamete forms, gamete being sperm or egg, from this genotype. And that would mean that we need to have 4 columns in our Punnett square for that individual. And so that's the law of independent assortment, that I need to account for the eye color and the wing shape, those alleles will independently assort rather than just say, oh, the dominants are always together or the recessives are always together. This is going to come up next week as well. But just to kind of a little primer of the two main laws that Mendel came up with, the law of segregation and the law of independent assortment. All right, and I think we got all the terminology we needed there. This last part, probability is kind of important to think about. It's going to help us set up our next lesson here. When we are okay with an event taking place, we don't care how the event, the order of the event. We have these two events that we want to take place, but we don't care about the order. That's going to cause us to add the two probabilities together. So let's say we want to get a boy and a girl out of our first two children. Well, what's the probability that we're going to have either a boy or a girl in that first generation, first child? It's 100%, right? And then what's the probability we will then get the opposite? Now that's going to be a little bit tricky. Because, well, if I had a girl, what's the probability I would then have a boy second? Well, now that's 50%, right? And then what's the probability that I would have a boy and then a girl? Well, that's 50%, right? So we have to then, once we get to that, we can start thinking about, I don't care how the first event takes place, because I just have to have a boy or a girl. And then I have to have the opposite, the second one. Well, it's 100% probability to start with. And then I just need to get the opposite for the second one. That's at 50%. So the probability of having a boy and a girl, then I would then bring in this rule of multiplication. I have to have the first event. I don't care which one takes place. Let's change how I'm going through this. I probably muddled this a little bit. There are two ways to get a boy and a girl. I can have a boy and then a girl, or I could have a girl and then a boy. Probability of getting a boy and then a girl would be 0.25, or sorry, 0.5 for getting a boy, and then 0.5 for getting a girl. The rule of multiplication says to go boy and then girl, I'm going to have that event, those two events take place. I would multiply those probabilities together. So 0.5 for getting a boy times 0.5 for getting a girl gets me to 0.25. So that first way of getting a boy and a girl would be 25%. But if I don't care about the order, if I just want to have a boy and a girl, I don't care which one comes first. I could have a girl and then a boy, and that would result in the same success that we put on this, or condition that we put on this for success. So again, it would be probably you get a girl, 0.5, probably you would then get a boy, also 0.5. Those two events would have to take place together. So we multiply those together, and that would be 0.25. So we have the first way that we could do it, which would be boy then girl, 25% chance of that. Second way we can get there, girl then boy, that's also 25%. Because we're okay with either path, we don't care which one gets us there, we can then add those two probabilities together. So the 25% from one and the 25% from two, we add those two together, and there's a 50% probability that we would end up with a boy and a girl. So that's kind of how these two laws of probability work together. Yeah. Questions on probability or any of the genetics terms that we talked about? All right, you're going to see how this is going to matter when we get to the next lesson. So, okay. So the first part of this lesson, we're kind of trying to answer this question here. Would you expect dominant phenotypes to increase, decrease, or remain constant across generations? It's pretty common for people to think that dominant phenotypes would become more prevalent. Well, because they're dominant, they show up even when they're heterozygous. So yeah, they probably should be more common over time. But what this lesson is trying to help us understand is that when we're thinking about evolution, if we think about it in terms of this population is actually a bucket of alleles, then maybe we can understand why dominant doesn't become more frequent. The idea is that our population just keeps reshuffling those alleles, but those alleles, even though those recessive alleles don't show up when they're heterozygous, the heterozygous allele that's there still gets passed on. Essentially, we just keep churning those alleles in different combinations, but they all still stay in the population. They're all still in the bucket. So anytime we go in and grab two, because two alleles is what makes an individual, right? Every time we go in there and grab two, as long as those two alleles get passed on to the next generation, those alleles are still, they're still there, right? So they don't go away. And so as we go through and look at these numbers, right, they're not the same every time, but from one generation to the next, we kind of see the numbers more or less stay pretty stable. If we were to do some statistics on this, we would see that these populations are not shifting, they're just slightly changing a little bit. And so it isn't that we're getting more dominant over time. In fact, if we start with 3 individuals who are homozygous recessive, 3 individuals who have white flowers, right? Everybody else has yellow flowers. Second generation, that goes up to 8. But it's back down to 3 in the third generation. We're not seeing large enough differences here to say that the population is shifting. Instead, we're just turning that same set of alleles over, and we're getting just some minor fluctuations in those numbers, just by random chance how they're paired together. And so we can see it probably best in this graph here, right? There's just not a big shift in the yellow phenotype over time. It's just, it goes down and then it goes back up, but it's pretty much constant. It's pretty average. And the white. You guys good with that idea that dominant alleles don't become more frequent over time, dominant phenotypes? Okay. All right. And it just gives us an example of some dominant alleles or dominant phenotypes that are fairly rare, right? That just kind of showing us real life examples of this not taking place. All right. The equations are going to be some key stuff here. Let's go ahead and slide this over here. We have 2 equations that make up our ability to analyze the evolution of genes. The first one is P plus Q equals 1. P represents the number of alleles that are dominant. So in our simulation, it was the yellow beads for the yellow flower allele. Q represents the percentage of the population that is, or the percentage of the alleles that are white, the recessive allele. So our equation essentially is saying is the percentage that is yellow plus the percentage that is white equals 100%. Right? We can reach into our bucket and pull out a yellow or pull out one bead. The probability that it will be yellow is the percentage of them that are yellow. The probability that I reach in and grab a white bead is the percentage that are white. And so the probability that I'll reach into my bucket and grab a bead that's either yellow or white is 100%. So we're adding those 2 probabilities together. So if 60% of our alleles were yellow, then we would say the probability that we reach in and grab a yellow is 0.6. And because that's 0.6, the probability that we get one that's white has to add up to be 1, right? So 0.6 and 0.4 equal 1. So that's our first equation. A major thing in working with these Hardy-Weinberg equations is understanding what information is being given to us or asked of us. Anytime they give you allele frequency, they're giving you either P or Q or both. If they are asking you to determine allele frequency, they are giving you, or they're asking you to determine P or Q or both. So whenever allele frequency is talked about, this is the equation that it's referring to. They're giving you one of those variables, or they're asking you to figure one of them out. Okay. The second equation is, instead of thinking of the alleles as a bucket, or instead of thinking of the population as a bucket of alleles, now we're thinking of them as actual individuals. Okay. P still means the probability of getting a yellow bead or dominant allele. Q still represents the probability of getting a white allele or white bead, recessive allele. But in this case, we're requiring them to be paired. So for P then, what's the probability that we'd reach into the bucket, grab a yellow bead, and then reach in and grab a second yellow bead? Well, in that case, those are two events that must occur together. And when there must occur events, we multiply their probability together. So if our probability of getting a yellow bead is 0.6, then we would take and say, well, what's the probability? Excuse me. What's the probability of getting a yellow and then another yellow? We have to multiply those two probabilities together. And so that's why we do the 0.6 times 0.6. So the probability of getting a homozygous dominant genotype is 0.36. Now we get to heterozygous. Well, to be heterozygous, we can reach into the bucket, pull out a yellow bead, and then pull out a white bead. That's a heterozygous individual, right? So we could take and multiply that together, 0.6 times 0.4. But when we're talking about being heterozygous, we're also okay with reaching in and grabbing a white and then grabbing a yellow. So there's two ways of ending up being heterozygous. We could go dominant then recessive, or recessive then dominant. So it's actually 0.6 times 0.4 plus, because we're okay with either pathway of getting there, 0.4 times 0.6. Because those are the same percentages, we don't have to do them 0.6 times 0.4, then 0.4 times 0.6, and then add them together. We can just use a little math and say we're going to times it by 2. So 0.6 times 0.4, because order doesn't matter in multiplication, right? 0.6 times 0.4, and then times it by 2, because there are two paths that we can get there. And so that's where the 2PQ comes from, is that those are the probability that we end up being heterozygous, with there being two ways of being heterozygous. Finally, Q squared, right? Q is the probability of getting a recessive allele. So reach into the bucket, grab a Q, or grab a white bead, reach into the bucket, grab a second white bead. So what's the probability that we do that? That's Y times Y, or Q times Q. That's where Q squared comes in. So we do 0.4 times 0.4, and that would give us the individuals. When we're talking about genotypes, we want to use the second equation in some form. If they're asking us, what's the probability that you would have an individual who's heterozygous if you have an allele frequency of 0.6? Well, in that case, I would just do 2 times P times Q. And then I could say, the probability you'd have an heterozygous individual is 48%, or 0.48. Okay. There's a second part to this, though. That is for genotype. But for phenotype, there are questions that are going to ask about that. What's the physical appearance? We could have the dominant phenotype. Remember, yellow was dominant and white was recessive. So you could have the dominant phenotype and the recessive phenotype. Well, we are still going to use this second equation for that, because the genotype determines the phenotype. So if we wanted to figure out how many individuals were going to be homozygous recessive, or sorry, how many individuals have the recessive phenotype, the only ones that will end up having the recessive phenotype are the ones that only have the recessive alleles, and that's the Q squared individuals. So once I know what Q squared is, then I know what percentage of the population will be the recessive phenotype. And if I know the recessive phenotype, I can figure out the dominant phenotype. So these are just how these problems are set up, right? Are they asking me about phenotype, genotype, or allele frequency? What have they given me? Have they given me phenotype, genotype, or allele frequency? And you can go from there. All right. Any questions on Hardy-Weinberg equations? All right, I forgot almost one of the key things here. Okay. Hardy-Weinberg are two different scientists. That wasn't the guy's name, although that would be a cool name. The reason why they came up with these equations is they wanted to be able to mathematically generate a prediction. So if I go and study a population of flowers and I measure, essentially, I figure out what their allele frequencies are. And then I come back 5 generations later and look at that population again. The assumption of Hardy-Weinberg is that no evolution should have been taking place for that flower population. The population is in Hardy-Weinberg equilibrium, that no evolution is taking place. Well, if that's the case, I should be able to say that within a statistical range, right, that based on the allele frequencies I measured 5 generations ago, I should see this many individuals with the recessive phenotype. The percentage of the population with this recessive phenotype, I should be able to predict that. And when I show up and I try and predict that, if the population, again, we'd have to run some statistics on this, is significantly deviated from the recessive phenotype prediction that I make based on the original allele frequency that I collected, that tells me that something happened. Something said, oh, let's say our recessive phenotype is higher than what we predicted. That tells me that there is some evolution taking place that is resulting in those with the white allele being favored over those with the yellow alleles. And as a result, they are reproducing at a higher rate. So when I show up 5 generations later, the white flowers are at a higher percentage than I would have predicted if there had been no evolution. And so this becomes a way to statistically show, quantitatively show that evolution has occurred. If a population is in Hardy-Weinberg equilibrium, there should be no statistical difference between the two populations, or sorry, between the 2 generations that I'm looking at. But if there is, now I have evidence that evolution has taken place. Now, once I have that evidence, then I can go and study the specific thing that might be taking place, right? Whether a predator was present, or a disease was there, or some other factor that was playing a role. Any questions about our Hardy-Weinberg equations lesson? Okay, that gets us to our quizzes. If you would like, you're welcome to jump off. You don't need to stick around for that. But if